Good morning and welcome to today's Potato Science Live webinar. Today we're covering biostimulants with Andy Cunningham. This is the penultimate webinar in this year's series. Tomorrow we're going to be looking at sustainability in potatoes. But before I hand over to Andy to talk about biostimulants, there's a little bit of housekeeping. So this webinar is being recorded by Syngenta. Nobody else has permission to record it. It'll be available to view on the Syngenta UK YouTube channel. If you've got any questions, please make use of the Q&A icon. Just click on there and ask your questions away, and we'll, if we've got time at the end, we'll do our best to answer them. We'll send an email out tomorrow with information of how to apply for basis and Naroso points. And for those that haven't joined this week, we can't see you or hear you, so you don't have to worry about that. Any of your faces being recorded. Okay, I'll hand over to Andy Cunningham. We'll make a start.
Morning all, how you doing? Hope you're doing well on this Thursday morning. Yeah, so thanks for joining me. I'm pretty much going solo today. I've been given the skeleton shift, so everyone else has had a partner. I'm going on my own today, so hopefully you don't mind putting up with me for the majority, but I am going to be your tour guide around biostimulants and everything hopefully you'll ever want to know. But I don't think it's a massive oversimplification, but bioproducts in general can be categorized into two groups. You have either biocontrols, which have a direct impact on biotic stresses like disease, weeds, and insects, but basically biostimulants don't, and they instead will stimulate a plant's natural processes to enable it to cope better with abiotic stresses. Now, the environment that crops are grown in can cause various abiotic stresses, the likes of at the top there, climate stress, so whether it's too warm or too cold. You then go down to crop quality, so certain stages of a crop's life cycle can be more tricky than others. So biostimulants can help improve plant processes during that stage. You've got nutrient efficiency uptake. Now, obviously with the price of nitrogen, it's very apt that this is an abiotic stress that we need to be concerned about, especially with relatively intense crop cycles. It can make crops more efficient at utilizing nutrients that are available to it. And this is obviously going to be more important going forward, but it's the climate stress that I particularly want to focus on this morning. Hope you don't mind. So just to set the scene, obviously summer 2021 was, I guess, a little bit disappointing, so we say, but nevertheless, our climate is changing, and it's not changing for the better. Basically, summers are going to get warmer and not in a good way, and it's predicted that they're going to be more frequent, and we're going to have more intense heat-related events. The likes of 2018 spring to mind, and they are going to be, apparently, to the Met Office, they're going to be 30 times more likely than they might otherwise have been. And if you look back over the last 20 years, 10 of the warmest years in the UK have been in that time. So you can see the trend is that heat is going to be an increasing important abiotic stress that crops have to deal with. Now, obviously, we're in the Potato Science Live webinars, so I think it's appropriate we talk about potatoes, and it's that heat stress that are particularly bad on potatoes, and it can affect how its natural processes occur. And basically, having a product like Qantas, which hopefully you're aware of, can help crops mitigate against it. But for those of you who've not come across Qantas, you're probably thinking, what on earth is that? Well, that's what I'm here to tell you about. So it's a product that's derived from sugarcane and yeast fermentation. It's actually inside it's got important nutrients such as organic carbon, potassium, calcium, and some amino acids. But it's with all this coupled together, it's going to help crops cope better with abiotic stresses. So it maintains them important processes that I've been talking about that might otherwise have caused yield loss. Now, what I want to do now is tell you exactly how it can help against abiotic stress. So we might have to go a little bit slow through this bit because there's a lot going on. But just starting off, I want to try and explain the Qantas benefit or the Qantas effect. And just to bring it back to almost GCSE biology, with photosynthesis. We've got a typical plant cell here. Modern potato varieties are adapted to grow between 14 to 22 degrees. And it's under them conditions that when plants utilize the light energy from the sun, you'll fix carbon dioxide and water into photosynthesis. And from that, it produces a sucrose that is then transported down into the tubers to help that bulking process. But you can have too much of a good thing. And when a plant is exposed to too much light and as a consequence, too much heat, the plant gets overexcited and it becomes stressed. What it then does, it'll then upregulate sort of photoprotective responses to protect its most vulnerable apparatus, which is the photosystem 2, which is in the chloroplast, them little green, them green things there, they're chloroplast. And inside there's what's called a photosystem 2. And that's the main driving force behind photosynthesis. So what plants do to protect it, they do what's called non-photochemical quenching. And this is a release of excessive energy from the sun. And rather using it in photosynthesis, it gives it out in heat instead. And I'll be using this non-photochemical quenching later, so you need to try and remember that. But basically what it does, it competes with itself. So it has to do one or the other. It almost struggles to do non-photochemical quenching while it's photosynthesizing at the same time. So in effect, it shuts down. So the more NPQ, the less photosynthesis. I hope that makes sense. And then what it then does to protect itself further still, it closes its stomata, so it reduces water loss. And then obviously that means there's less CO2 coming in, so there's no photosynthesis going on. Now, just to complicate it a little bit further, the plant will have what's called reactive oxygen species in there, otherwise known as ROS. Now, plants constantly produce ROS as a byproduct of photosynthesis and other respiratory actions within the plant cells, but they also act as a stress signal to the plant. Now, under normal conditions, it's not a problem, and a plant will release antioxidant enzymes that can deal with them. So we'll get rid of them, ROS, but it is under that excessive stress that a plant just can't cope, and the ROS levels just get out of hand. So we end up with increased levels of ROS. And when there's an overproduction of these, the plant just can't cope, and it exceeds the plant's natural defense to deal with them. So these ROS end up snatching electrons from essential organic molecules and disturbing normal cellular function. So you've got a double whammy. You've got reduced photosynthesis, and then you've also got that overproduction of ROS. So at best, the plant is shutting down, it's not producing yield while it's under that stress. But at worst, it can lead to cell damage and ultimately affecting the plant going forward, even when that stress has then passed. So this is where Qantas comes in. So dealing with that ROS is very important, and throwing Qantas into the mix will allow a plant to overcome those issues. So getting in there before the plant is already stressed will give it the best chance to cope with that heat event, which will be important when I tell you how we recommend the application of Qantas. But how does it work? So firstly, it works as an antioxidant. So it upregulates them antioxidant enzymes I was telling you about, so the likes of what's called catalase or glutathione reductase. And this basically is an immune system. So it boosts the immune system to deal with them excessive ROS. Secondly, I mentioned that in Qantas, there's a high portion of potassium, and that you have a high concentration of potassium, it gets inside the guard cells. And if you've got high proportion of what K+ ions, it makes them more hypertonic, and more water is drawn into the guard cells, keeping them more turgid, and so widening that stomatal opening. And because of, obviously, if it's open, the stomata is open, as I mentioned, when it's stressed, when it's closed, when it's open, it's able to take in more CO2 and it's able to transpire. So it's allowing photosynthesis to carry on. I hope this is all making sense. I hope I'm not going too fast. You'll be pleased to know that it is being recorded, as Gus mentioned, so you can re-watch this if you might need to go a bit slower. But basically, both of these functions help the plant to maintain healthy cell function and to optimize photosynthesis in the cells. So the more light that's being used in photosynthesis. Now then, we'll move on from that and we'll go a little bit on to what temperature is important on potato development. Obviously, I mentioned between 14 and 22 degrees. 14 and 22 degrees is the optimum temperatures for potato crop, but it will depend at which stage the crop is at which temperature it will be optimal at. And when you look at sort of optimum temperatures for photosynthesis and around that tuber initiation time and tuber bulking and tuber yield, any temperatures over 25 degrees, it will struggle, right? So how do we show that Qantas is helping? Well, we go to some learned people at Nottingham University. Now, this is the sort of package that I hope will help differentiate Syngenta from the rest of the biostimulants on the market. Nottingham University did this nice bit of work to help justify what Qantas is doing in the plant. Now, however, right, okay, if you want to hear more from this rather than myself, from the actual scientists that have done it, we've actually done a video, and I think Kirsty, who's on this call, is going to put a link into the chat to the video if you want to watch that at a later point. Hopefully not now, because yeah, you won't hear me talking, but you'll also get a link to the video that'll be emailed out with this recording, and it's also going to be on the website, so hopefully you won't miss that. So that's Dr. Rumiana Ray. She's a professor at Nottingham University who's helped organize and carry out this work for that I'll hopefully do justice to you now. So in summary, I'll just summarize it now. They monitored a stressed potato plant over a 7-day period, where that heat stress was applied 24 hours after an application of Qantas at 2 litres per hectare. And obviously the aim of the study to find out exactly what Qantas is getting up to inside the plant, not only looking at plant physiology, the likes of photosynthesis, but then digging further down to that hormone level to find out exactly what Qantas is getting up to inside that plant. Now, this could get a little bit heavy again, so I'll try and go relatively slow, but not too slow. But just as a reminder, it has obviously been recorded, so it doesn't matter too much if I lose you at some point. But they stressed the plant to 30 degrees. Now you're probably saying, well, that's not 25 degrees that you said, Andy. Well, this is in a glass house, so it's not in the field. And they wanted to make sure that they stressed the plant to a certain amount so that they would be sure they were going to get a response. So that's why it was 30 degrees. Now, I mentioned that when a plant is photosynthesizing, carbon dioxide moves from the air through the stomata and into the plant, and then water vapor will then transpire and diffuse out of the stomata. Now, under that heat stress, they were able to demonstrate that the Qantas treated plant was using more CO2. And also it had increased levels of transpiration, which shows that the stomata is open and the plant is actively photosynthesizing, which is a good start. Now, if you remember, I mentioned about non-photochemical quenching, so NPQ, and this is that quantifiable measurement of unused light that the plant's getting from the sun and then turning it into heat instead. Now, using what's called a fluorocam, they were able to produce these images and create this quantifiable data that showed that the Qantas treated plant had a significant reduction of NPQ, and that's exactly what it wants to be doing, because it's not protecting itself, it's carrying on photosynthesizing. Now, how do I know it's carrying on photosynthesizing? Well, we look at what's called quantum yield. Now, this is a measure of converting the sunlight into the energy through photosynthesis, because when plants photosynthesize, they'll emit back certain frequencies of light known as chlorophyll fluorescence. So the more fluorescence, the more photosynthesis. So you can see again on that chart, the Qantas treatment plant is having a significant increase in quantum yield. And even when you look at again, them fluorocam images on the left-hand side, you can quite plainly see the Qantas treated leaves were significantly redder, if that's even a word, than the control plant. And the more red, the higher the quantum yield, which is what we want to be seeing. Now, I mentioned about we're going to go down into the hormone level, and hopefully you will take this as it is, because we want to show that Qantas is having effect on the plant, and we can show that it's having effect both in terms of the physiology, but also down to the hormone level. Now, these two hormones are heavily involved in reallocating and stimulates to and from the tuber. So under normal circumstances, gibberellic acid on the left-hand side is a hormone that's involved in promoting cell and stem elongation, but it's also involved in inhibiting tuber formation. So before we get to tuber initiation stage, gibberellic acids are quite high because they want to promote foliage growth and everything around that to create photosynthesis in the leaves. When you do get to tuber initiation, you'll find that gibberellic acids are decreased, and then the cytokinin levels on the right-hand side, well, they increase, because you can see that they promote cell division and expansion. They actually inhibit cell elongation, so they are known to promote tuber formation. So you can see that this hormone in particular, the cytokinins, is massively important to tuber development, meaning that energy is then shifted from the foliage down into the tubers. So the study then showed that the stressed plant went into survival mode, basically, and it started protecting itself. The cytokinin levels, you can see on the left-hand side, were reduced in the blue bar, and on the right-hand side, the gibberellic acid went up. So it started sending assimilates back into the upper growth to try and protect itself because of that heat stress. With Qantas, because it's not stressed, it was able to maintain its normal functions. So you can see that in the Qantas treatment plant, the cytokinins were significantly increased. And then quite nicely, the gibberellic acid was lower. So you can see that it's working as it would otherwise would be doing. But does this translate to yield? Yes, is the answer. Plants were taken to yield, and they showed that they had a significant increase in tuber weight, which was really good. But importantly, when you look at the distribution of them tubers, there was a significant increase in them, the larger grades that you would want to be getting, rather than the smaller grades is what you don't want. So that's all good. And we've shown, basically, just to summarize, we showed in the lab that Qantas is having a significant impact on how the plant is able to carry on working. So we've looked at the physiological phenotyping. So we've got lower MPQ, we've increased photochemical efficiency and higher photosynthesis, which is all good. We've then gone down into that molecular level, and we can show that we are having an impact on both the cytokinins and the gibberellic acid. And then we've also shown that we've increased tuber weight size. So that's a really good start, and really sets the basis for not only in potatoes, but a lot of crops that behave very similar to potatoes. But how does it work in the field? Now, if you remember my colleague Michael Tate, he presented this at last year's Potato Science Live, I think. So we did a large number of split field work in 2020, and ADAS did this work anyway, and they did, out of the 32 large split field trials, they showed that there was a significant increase in yields where Qantas was made before a heat event of 25 degrees or more. And also you can see even on the extreme, although it's not significant, it's so close, it's ridiculous. So you can see that the trend is that an application of Qantas before a heat event is having a significant effect. And that's what we want to be able to show, is that not only is it working in the lab, but it works in the field as well. And this, I guess, is a bit of a shameless plug again. I did record a video of another split field that I did this summer. And if you want to watch it, Kirsty, I think, is going to share another link in the chat, which will take you to our YouTube channel to go and have a look at that. Basically, it was a trial I did at the whole crop marketing demo site in East Yorkshire, and I won't tell you too much about it, so hopefully you go and have a look at it, but it demonstrates what we want to be seeing from Qantas. But rather than looking at videos, I thought I'd tell you some other work, just because the more work that you see, hopefully the more confidence that you get that Qantas is behaving how we want it to do in the field. So we tried it out in Scotland, and it's possibly not the first place you'd associate with heat stress, but it turns out they had a really decent summer up there. I'm down in Doncaster, so it's up there. And this was a split field trial applied according to our 2021 advice, which was an application at tuber initiation, and then 2 subsequent applications at fortnightly intervals. So 3 applications of Qantas. Now, I did mention earlier about how that tuber initiation time is very sensitive for potatoes. So protecting them at this stage is very important, and will probably remain part of our advice going forward. But we'll come on to my sort of recommendation around Qantas and how it's evolving the more we're learning about Qantas later. But just to focus on this site in particular, you can have a look at the weather that we see, and I've put that red line in, which is that 25-degree trigger point for potatoes and heat stress. The arrows at the bottom are where it was sustained 25 degrees. So it was for 4 hours in that day as well. So that would just be a sustained heat event. But then the parameter of our heat events would be that if you had these 25 degrees for 3 days in a row as well. Now you can see that the ones in yellow are where that was achieved. Now I mentioned on the previous slide that the applications were made to our current advice, which is tuber initiation, and then 2 more applications after that. And you can see that when I bring them in, and we have around a 14-day protection period with Qantas, you can see that they covered them heat events quite nicely, and quite fortunate for me, which means that when we go to you can actually half expect to see. So 3 different areas from treated and untreated, basically, and there was 18 plants. And what you can see from the number of per grade size, it actually looks the wrong way around. More tubers in that untreated than the Qantas is that below 45 mil. But that's because the treated plant was actually carrying on bulking its yield, which is why when you get to the larger grades, you've got more of there. So when I bring in the actual total, it like I said, it looks the wrong way around. You say, well, you treated plants giving away less tubers. Well, that's because they're all where you want them to be. They're in them bigger grades, and that's bore out by the weight of each of them grades as well. And quite nicely, we've ended up with 6 kilos more in them digs that they did. If you multiply that up to field scale, that equates to nearly 14 tons increase on a 55-ton crop, which you could imagine more than pays for the Qantas that was applied to there. If we go across to the Netherlands, they had a large number of split field trials this year. Our Dutch colleagues did a nice bit of work, and they had 27 trials, in fact. And it turns out they have a similar climate to us, especially last season. They only had 1 heat parameter again. The 2020 season was a bit tricky, wasn't it? And basically, they carried out 2 different treatments. It wasn't just treated, untreated. There was one that was carried out to our current advice, which is 3 applications at then fixed timings, or an application as and when a heat event occurred. Now, as it turned out, there was 1 heat event, so there was 1 application in total. And 100% of them 27 growers applied it at the tube initiation stage, because that's when the heat event occurred. Now, if you just look at the yield, you can see that there's a 9% increase with them fixed timing. So when you compare that to the advised time, there is actually a slight improvement. But if you remember, there's 3 applications of Qantas there versus the 1 with the advised timing. And if we bring in the size distribution, it looks to have improved the grades throughout, even in them lower grades. But 35 to 50 mil is not stupidly small, I wouldn't say, so that's not a problem. But even at that 50 to 70 and the 70 plus, there's still more in there. So the trend is, again, although I haven't got any stats on this data though, is that you apply Qantas at the right time, you'll get an increase in yield. So if you assume, again, a 55-ton crop, that 9% increase is going to be a 10-ton increase per hectare, and then that 7% increase, again, it's only a ton less with 9 tons over the untreated. So here we are, the relative end of my presentation. Just a few take-home messages, I thought. I just wanted to not sort of hammer home, but just as a reminder, climate is changing. Them heat waves of 2018, 30 times more likely. So we need to be protecting our crops. And using biostimulants is something that is probably new to a lot of people, but so is climate change and the way that our summers are going. It's something we need to be aware of now. And heat stress is significantly affecting crops like potatoes, but Qantas has been shown to help mitigate that heat stress to help crops achieve their maximum yield. So where do we go next season? We're looking to have a hybrid advice, so carrying on with that tuber initiation time. As I said, it's very sensitive time for potatoes. But we're going to look towards maybe we are looking into having a prediction tool where we can predict. I know it's hard. I know it's difficult to predict weather half the time, but we've got some stats that would suggest that we can predict relatively accurately from 5 to 7 days out when a heat event is going to occur, and hopefully that will give you alerts to basically say, right, you can put Qantas on now. You know, we want to be in a position where you say you don't need to put Qantas on yet because you've not had them heat events, but then you get an alert when you've got a heat event coming, put Qantas on. And I think that is a new stance where biostimulants are, rather than just hopefully sticking in the tank and hoping for the best. We're in a position where we can predict quite accurately that Qantas is going to bring a benefit. And we're planning to do some ground proofing with this. We're around 20, 30 growers this coming season. But other than that, I think I'm done. And if you've got any questions, I'll try and answer them. That's right.
Okay, thanks very much, Andy. That was a brilliant presentation, very informative. We've got a few questions.
Okay.
First one, how does Qantas compare to competitor biostimulants in potatoes?
Oh, that is a question I always do get asked. Good question. The thing about biostimulants is that they'll always have different recommendations or different areas that they'll say they major in. So what we wanted to do with Qantas was completely understand what it's doing. So we put all our resources into knowing what Qantas is doing and when best to apply it. Now, that might not be the case for brand X biostimulant. So in some instances, it probably would be unfair. Maybe it's an easy get out for me to say this, but unfair to compare Qantas with another biostimulant, because that might not work the same way that Qantas does. We wanted to make sure that we knew how Qantas worked so that we could recommend it quite confidently and be in a position to say, if you put it on now, you will see a benefit. It will not be money lost with Qantas, it'll be money gained. So that's a long-winded way of saying I haven't really compared it with other biostimulants yet. It's probably something that we probably need to do, but when we're bringing a biostimulant to market, we wanted to make sure that we knew how this product worked better than anything else that's out there at the minute. But that probably does mean that it's not being compared with other biostimulants. But yeah, I guess that's my answer for now. When we get further down the line and we know exactly what Qantas is doing and how it, we've got no more questions to answer, maybe we can go further down that road and see how they compare with other products.
Okay, and we've got someone asking a question about salad potatoes. Is that something that we've done any work with before, salad potatoes, or is that something that might be coming in the future?
It's not something we specifically looked at. We haven't really targeted any, whether it's ware, salad, seed. We just kind of, in general, looked at the broad scope of potatoes. It's something we probably could do with focusing on. Looking at the data, it would suggest that we're helping tuber initiation rather than halting it. So I can only imagine it's going to bring a benefit, but it's not something I've done trials on currently. But it is something that probably could be done.
Maybe we could take that one a bit further offline with that customer.
Yeah,
so
absolutely.
They were. Yeah, it's quite a specific question. Okay, another question: how does Qantas compare to on other crops other than potatoes?
Right. Okay. It's funny you say that. Actually, do you mind if I try and dig something out? Because
yeah, that's fine. Yeah.
Right. Sorry. Right. Let me get this. Where's that gone? Right. It's all very professional. Sorry. Let me do this and that. And am I meant to? Yeah. Share screen. Can I take control? That and that. Right. Let me know if you can see that.
Yes, we
can. Can you?
Yep.
you seeing some sugar beet?
Yes, we are. Yeah.
Ah, right, nice. I'll put that in presenter mode so it looks relatively presentable. So I did some work on sugar beet last season, and there was a lot of done in split field work, which again might not be overly convincing to some people, but we did also look at some replicated work, and this was some work we did with GCI. They've done some nice work with us on potatoes, so I thought, well, I'll treat them to some work with sugar beet as well. And you can see that when we had early applications of Qantas, there are stresses that come with sugar beet, both in terms of herbicide stress and drought stress and things like that. And we were seeing that maybe towards the back end, you were getting a nice increase in yield when Qantas was applied. We've also looked at Megafol, which is a Valagro product, and that looks to perform quite well. And that's a seaweed-based biostimulant, which might be actually ideally suited towards this, because seaweed-based biostimulants are ideally positioned to help with establishment of crops. So that is exciting. We also looked at Qantas late on, and similarly with that heat stress, you can see that I've timed these. First application you can see is early root expansion. I did originally think I'll just look at root expansion, and then each time when the fungicide is to be applied. And the thing is about when fungicides go on, you can't dictate when that's going to go on. Now, when we did this, we had that heat event in July, as I told you, in the potatoes, and I said to him, right, okay, let's have a targeted application as well. So you can see that application B is applied just before that heat event, and you can see that we're getting a significant increase in yield, and that is significant. And that's plot work, which hopefully then gives you confidence that the split field work is actually true, because we did a trial in Lincolnshire as well. You can see the blue line is where Qantas was sprayed, and the oranges obviously didn't get Qantas at all. You look across to what's that middle bit? Biomass. You can see where the Qantas is got more biomass, and then the water usage, it also looks to be using more water as well. And just looking at the weather, it's almost like I prepared this presentation. I feel like I knew you were going to ask this one. It wasn't as stressy. You see that 25-degree line that I used for potatoes. You bring in when that application was applied. It should have really been applied earlier, to be fair. But this is applied when the grower had a chance to go in, and it did cover that heat event that would have triggered a potato heat event. And then when you look at where I did the digs, this is what I did. So I tried to be as fair as I could do. So it went pretty close to where that split was. You can see on that image, hopefully, the Qantas treated plants or roots are significantly larger. And then when I weighed that, with my dig, with my fork, should I say, you can see we're almost getting a 37% increase in yield. And even if we got half of that benefit, again, Qantas that was one application of Qantas before that. So really quite impressive. And in fact, we are going to get sugar beet put on the label. It's one of the benefits of biostimulants is that you can pretty much go put onto most crops without having to register it. But I think our stance is we want to prove that it works on these crops before we put them on the label. Now, this work that I've done with GCI and obviously the split field work, hopefully gives growers and agronomists the confidence to say, yeah, Qantas is something I want to put on my sugar beet. And so that's where we're at with that. So that was a long-winded question to say sugar beet is something we're looking at, and also maize is something we're looking at. Obviously, herbicide stress early on, and then around that tasseling time is probably where we're going to be looking to target that, and also spring crops, spring cereals.
Okay, just to get you back on potatoes then, Andy.
Oh, sorry. Yeah.
We've had a few questions around water, and people wanting to know what a tight irrigation program, what effect that would have on Qantas, or have we looked at any trials where there's been irrigation and no irrigation? Yeah.
So we have seen the same benefits in irrigated crops. obviously the thing with irrigation is you do get the cold shock as well. It's not just, but you are reducing the temperature for that short period of time. So there are elements of reducing stress. So it's probably not as stressed as it might otherwise be if it wasn't irrigated, but we are still seeing them same benefits in irrigated crops as well as in unirrigated. And like I say, irrigating crops does bring an element of stress in itself because of the cold shock. So whether or not Qantas would help with cold shock stress, I don't know, but we are still seeing the same benefits in irrigated crops as well.
Okay, and then probably a final question to close on. Any work done on cold, wet conditions we have in Northern Ireland?
They are looking at Qantas in Northern Ireland. Whether Qantas is the product, obviously we are looking at it on heat stress, which seems like complete opposite of that, but it is helping with biotic stresses, as I mentioned. We do have a full range of biostimulants as well. You don't just have Qantas. We have like Megafol, which is ideally suited, I would have thought, to them sort of conditions. So cold stress, Megafol would probably be biostimulant that I would probably recommend in that scenario.
Okay, great. Right, well, thank you very much for everybody participating today, and thank you very much to Andy for a brilliant presentation. So before we close, a reminder tomorrow that we've got sustainability in potatoes, and just a thank you again.
Now, thank you all for joining, and hopefully I'll see you soon.
Thank you. Bye.
Cheers. Bye.